Welcome, everyone. My name is Bernadette, and I'm here organizing this symposium with Adam Jones, and we're very excited to have you all here. For our first speaker, I would like to introduce Alberto Corral Lopez.
Thank you, Bernadette and Adam, for organizing this interesting symposium. I will be talking about neural mechanisms of my preference in this piece, the GAPI. So I'm moving directly to show you some early work where we developed GAPI selection lines with difference in brain size, and with this, with around a 40% difference in brain size after 3 generations, that came with certain associated difference in neuron numbers and cognitive ability. And we, in this investigation, we use these lines to study the link between brain size and mate choice. Specifically, using dichotomous choice tests, we quantify preference for colorful males in these females, a trait that is quite well known to be a reliable indicator of male quality in this species. So our results in this experiment show a significant preference for attractive males in large brain and wild-type females, measured with time associating with this type of males as a proxy. However, we did not observe such a preference for these males, these colorful males in small brain females. So that led us to further work using transcriptomics to evaluate what happens in the brains of these females that actually show a preference for the colorful or this attractive male. So in this work I'm mentioning, we expose preference and non-preference females to 3 different types of stimuli: an attractive male, adult male, and also to a female for associate as a measure of a social interaction with a conspecific. And then after this time, we collected brain samples for transcriptomic work from 2 different regions of the brain of these fish. The optic tectum, highlighted here in yellow, where is known to be involved in the sensory processing of visual signals in teleost fish. Also, from the telencephalon, you can see which is highlighted there in red, where this information is integrated and decision-making takes place in the brain of this fish. So with this experimental setup I just showed you, we could, for instance, obtain a series of genes that are differentially expressed in the telencephalon and the optic tectum when these fish were exposed to these different social stimuli. And by evaluating, for instance, overall expression patterns in these differentially expressed genes by means of principal component analysis, we observed that, for instance, preference females exposed to a colorful male show a unique transcriptional signature, as you can see highlighted in these figures. Also, as highlighted now, we also observe a quite stronger differentiation of these categories. So preference females exposed to attractive males, but only in the telencephalon, not in the optic tectum, so in the telencephalon where the decision-making takes place. So overall, these investigations highlighted with selection lines, with GAPI selection lines, and using transcriptomics highlighted that brain size and correlated traits like neuron size, neuron number, or cognitive ability are important mediators in mating context. And further functional analysis that I didn't show today within this work allow us to provide further depth into neural mechanisms of decision-making in mate choice, and also to provide evidence for the remarkable degree of flexibility that mating decisions can elicit in the transcriptional network of the brain. But I went up. No. So, of course, when it comes to behavioral decisions, I guess as many other biological traits, the norm is that a trait such as preference is not stationary across contexts, as I represented from this graph that you cannot find probably in any paper. But more or less, rather the data that we expect is that behavior will be very, very variable among contexts, like in this other plot I made up. So our current work using GAPPIs involves incorporating context-dependent choices that drive mating decisions, and then with this assess neural mechanisms driving preference in this piece. Okay, I'll take some water. So next, I will talk about with this concept, I will talk about the progress in two projects that incorporate these context-dependent preferences. First, in females, and characterizing transcriptional response of female GAPI mating preference at different reproductive statuses. Also, using males, we're using these concepts to characterize transcriptional response of male GAPI mating decisions associated with prior experiences in these males. So context-specific decisions can happen. So starting with our project in the female, we know from early work in this species that while females can accumulate male sperm in their gonopore, during the whole reproductive cycle, actually, we know that during the reproductive cycle, they actually reduce strongly the frequency of sexual behavior after the 2-3 days following parturition. As you can see in this graph from early work on this species. So with this in mind, we designed a project to assess preference for better quality males or more colorful males. So following at least the experimental setup that we came up for previous projects. And we assessed this preference under 3 different reproductive statuses. We had virgin females. We also have receptive females, so females that were taken 1 day after parturition, and we also have non-receptive females, so 14 days into their cycle after parturition. So some results from this test, using time associating with the different males again, a similar approach. As a proxy of preference, we see that only receptive females show preference towards colorful males, which is adding new information to these earlier studies in sexual behavior response. We see not only sexual behavior change, but also preference toward better quality males during these early stages after parturition. So again, we could now assess neurogenomic responses associated to these optimal behavioral decisions in the females, for females that we used. And for that, we replicated our design, exposed receptive and non-receptive females to colorful, attractive, and to dull males. And then we, again, collected tissue from telencephalon and optic tectum to create these replicated pools of tissue for each of these 4 treatments. So we proceeded to perform a routine differential spatial analysis to capture genes that are differentially expressed across these evaluations of colorful and dull males. And as you can see here, we observe relatively high numbers of differentially expressed genes, but very, very little overlap in these genes that are differentially expressed in receptive and in non-receptive females. So interestingly, we find this approximate 2-fold increase in number of differentially expressed genes between these 2 reproductive statuses, with what I found very interesting, that was higher numbers in the telencephalon or receptive females, again, which are making more optimal decisions, as we recognize. But we see this opposite pattern in the optic tectum, where this approximate 2-fold increase in the non-receptive females. So looking at overall expression patterns of these differentially expressed genes, we indeed complete a complete separation of reproductive status and type of male samples across both tissues, which to me is an indicative of a unique transcriptional response for all of these conditions in both of these treatments. So they are completely activating different gene networks in the brain. So moving into the second experiment, focus on male decision making, we created what we call experienced and naive males by housing them or by not housing them with females, which allow them to experience or not to experience females at different stages of their reproductive cycle. So next, after we obtained these males from this treatment, we proceeded to perform quantification of male behavior in those 4 conditions. So this 2x2 factorial approach. And specifically, we focus on 2 major behaviors that usually are quantified in a GAPI. We have sigmoid displays, in which males show off their coloration towards the female. And from previous studies, we know that offer a higher rate of success in sperm transfer than this other second behavior, which is the denominated SNCC attempts, in which males attempts to inseminate females without consent by sneaking from behind and inseminating them. So from an optimal decision-making point of view, one will have the expectation that males that have been exposed to females and have this experience will increase the rate of display towards the receptive females, but decrease the rate of display towards the non-receptive females. The other way around will be the expectation for these for coercive behaviors. Well, you will speak that experienced males will increase their SNCC rate towards non-receptive females. And we found a lot of variation in these behaviors, not very strong effects, but those effects that we found, they're actually pointing toward these directions. We have experienced males, but not naive ones, increasing the rate of displays toward receptive females. And then we have experienced males performing more SNCCs towards non-receptive females. So we use this variation in individual behavior of the males within our treatments and then we calculate the classical measure of behavioral performance, which is a ratio of the display versus SNCC, and then we take advantage of new developments in differential spatial analysis, where now we can correlate individual behavioral variation to transcriptomic profiles or individual samples. And actually, we collected these samples precisely after the experiments in these males, and we can perform this correlation analysis where we find a series of differentially expressed genes associated with either an increase in display rate or an increase in SNCC rate. And what we see is that across these treatments, we find a clear increase in the number of differentially expressed genes in the telencephalon of experienced males when taking optimal decisions, as represented here with this patterning, which is highlighting displaying more toward receptive females or SNCCing more towards non-receptive females. Have increasing in this treatment. So I'm going to stop here now. Hope that soon I will be able to present you further insights in functional analysis of these differentially expressed genes that we find, also co-expression gene networks in relation to these results, and also comparison, we're aiming to compare gene regulation of these gene networks, which are associated to optimal decision making in a mating context across sexes, which are analyses that are still ongoing. So this work has been, with that finished, and this work has been developed at different stages of my career, and in the meantime I embark in many other scientific and personal projects, but one constant was great support I have and advice I have from Judy Mank. Also, I learned a lot from my colleagues in all these projects and in previous early works. Versada, Wouter, Julia, also former collaborators from Stockholm University. So thank you so much. I went a bit long.
All right, Right, thanks so much for inviting me to this wonderful symposium. So my name is Wouter. I'm a postdoc with Judith Menk at UBC. And I'm going to continue on the Guppy theme, but now less about decision making and mate choice specifically, but more about the traits that the mate choice is on, what they are actually choosing about. All right, so let's jump in. Yeah, this must work. Okay. So Alberto just, I think, convinced us all that the guppies are a really wonderful system to study mate choice and sexual selection, and guppies have been a very successful system in this regard for many years now. But I think the main thing that drove interest into the system in the first place was that the males have these extremely variable color patterns. So the males I'm showing here and on the side, they're all from a single lab population. You can basically find these in one tank in the lab. And you can see each male has a more or less unique combination of spots and stripes of different colors. And we've known for almost a century now that these tend to be very heritable and that they are partially sex-linked. However, somewhat surprisingly, even in the current genomic revolution, we actually don't know very much at all about what are the genomic loci that underlie all this variation. And personally, I think this is a bit of a shame, because I think this could really help us to answer some central questions in the system. So for example, a main question a lot of people are interested in is how is all this variation actually being maintained? And so just to illustrate how magic guppies can sometimes be, there was a really nice paper last year in Science that showed that even in a small population of only about 300 males, there's 27 different Y haplotypes that are being maintained by selection somehow in this system. So the hope is then that we're going to try to find some pattern loci and then return to the field and actually be able to study some of these loci in natural populations. And for example, look for evidence of selection in these loci. And that we can learn something about the genetic mechanisms that underlie all this extreme polymorphism. All right, so the approach that we took is basically to breed as many fish as I could possibly fit into the lab. And so we used this pedigree design where we, at the same time, do sort of artificial selection. So we basically breed a lot of fish, and then we photograph every single male. And then I both up-selected and down-selected on the amount of orange that these males have. And we basically did this whole design 3 times, so 3 independent replicants. And so to jump right into the data, the selection is very effective. So here I'm starting at the parental generation and cascading down to the F3s. And you can quickly see the up and down selected populations diverge and how much color they have. The thin lines here are just connecting fathers to sons, so they're showing patrolines. And we can see after only 3 generations of selection, the up-selected males are already twice as orange as the down-selected males. And just to show you some of the extremes that we have at the end, we have males that have basically almost lost all orange coloration, and we have males that really have these fat chunky spots that females really like. All right, but this is a very gross measure, this overall orange. And one of the exciting things about all this variation is that there's all these different spots and all these different things going on. And so we really want to do a much more thorough job of phenotyping these fish. And so for that, I developed a bunch of deep learning methods to basically try to analyze all these photos. So we have about 3,200 males in the experiment and about 14,000 photos in total. And so that's why these kind of AI approaches are extremely helpful. And so we basically first extract the fish body from the background. We then place automated landmarks all across the fish. And then we can use geometric morphometrics to perfectly align all the fish to a common reference shape. And then we can also just extract all these color ornaments that we're very interested in. And so we did both orange and black. I'm just going to talk about orange today, but if you're interested in black, it's all in the preprint, or come talk to me later. And so what we end up with then is all these really nicely aligned patterns of all these different males. And then now the question is basically, can we actually figure out what's going on with all these colors? What is actually heritable? It seems like overwhelming much variation, right? So, okay, so some of the things we can do is we can then, since we have nicely overlaid all these fish, we can sort of do these pixel by pixel comparisons at every location of the body. And so the first thing we can do is just can look where is the orange occurring, right? So this is just the guppy body. And all the gray parts is where only less than 1% of the males is orange. And then the colored bits is where there's more common orange. So we have really bright spots where almost everyone is orange, but then most of this is actually orange is relatively rare, and that's where all the uniqueness of these patterns comes from. Then we can look at what actually happened throughout selection. So this is just a F3 comparison between up and down selected males. And we can first see that we basically see color all across this plot, which means that the selection basically affected orange all across the body. So it's not like we just selected or 1 or 2 ornaments, but everywhere in the body we got a selection response within 3 generations. And very interestingly, the red areas here are in the expected direction. So the up-selected males are more likely to be orange there. But we also see the interesting blue areas where we see the reverse effect. So the up-selected males are less orange there, which tells us there's genetic correlations between some of these ornaments, right? But then what I was personally most excited about is that we can then combine this with our pedigree information, and we can do the quantitative genetic analysis again across the body, right? So here I'm just plotting the heritability of being orange, yes or no, at every part of the body, and we can see that this orange coloration is really locally heritable. So we see all these brightly yellowed areas, the heritability is really close to 1, but even in the tail here, where there's the less heritable areas, that's still a heritability of about 0.5, right? So really locally heritable. And then we can actually separate out this heritability into the different components, so both the autosomal, X-linked, and Y-linked variation. And we can see there's a sort of diffuse pattern of autosomal variation all across the body. And then there's particular ornaments that really seem to be strongly sex-linked. So here we have this X-linked spot in the tail that really pops out. And here's an example of a male that has that particular X-linked bar in the tail. And we also have these colors here at the front, near the gills and the pectoral fin, that really show this very clear Y-linked pattern. And here's some males that have some of these color spots. All right. So, so far the phenotypes. Let's actually look at the genotypes now. So we took about 300 males of the F3 generation. We did whole genome sequencing, and then we did GWAS on the orange pattern as a whole. And so this is a Manhattan plot. If you're not very familiar, I'm just plotting all the different chromosomes. And then on the y-axis, so each spot is a SNP or a small indel. And then on the y-axis, I'm just plotting how strong this association is with orange pattern. And you can see there's a whole lot going on, but there's 2 things I want to highlight. So the first, we see this huge elevation all on chromosome 12. That's actually not very surprising because it's a sex chromosome. And I just showed you there's a bunch of sex-linked effects, right? So that makes sense. But we also see a peak basically on every autosome, clearly indicating that these color patterns, they're really highly polygenic, right? All right. So underneath each of these peaks is a story, but I don't have time for all of those, so let's just focus on one of those stories. And if we're going to look at one, why not look at the tallest one, right? Good place to start. And this is on chromosome 14, an autosome. All right, so what is actually going on? Like, what is this actually encoding? So we can just look at actually the phenotype that this seems to be very strongly related to, which is this spot near the pectoral fin. And we can see that the homozygotes here, they never have this spot, and the heterozygotes almost always have this spot, right? But there's 2 curious things that are going on. First of all, this is a Y-linked spot I just showed you, and this is on chromosome 14, which is not the sex chromosome. Okay, that's odd, right? The second thing is that this other homozygote is completely missing. We don't have any individuals that have that. Right, so this is a non-Mendelian site. So what is actually going on? Well, one hint is that if we just look at the allele counts there, we can see that these homozygotes, they have the expected 2 reference alleles, but these heterozygotes, they also have those 2 alleles, and then they have a bunch of extra reads that are mapping there from somewhere else, right? So this suggests to us that this is some kind of copy number variant, and it suggests that it's Y-linked, right, because of the phenotype relation. So we can come up with a hypothesis. What do we think is actually going on? Well, we think there's some kind of region on chromosome 14, maybe, that has been duplicated to the Y chromosome into some kind of region that is not in our reference. And then when we actually try to map all these reads, they all map to this place in chromosome 14. And we basically get this stack of extra reads that are not actually from chromosome 14, they're actually from the Y chromosome, right? And we can confirm that if we just look a bit closer into this region. So here, I'm just zooming in on this region, and we can see there's one gene here called texin. We also know that there's a helitron here, which we think might be involved in actually copying this gene to other places in the genome. And what I'm plotting here is the coverage in this region, comparing in black the male coverage to the female coverage in red. And we see that the female coverage on a lot of this is much lower than of males, and that again suggests that this is indeed coming from the Y, right? And then finally, if we just look at all the variants in this region, so all the SNPs and small indels, we can actually use our pedigree data to see whether these fit a Y-linked inheritance or an X-linked inheritance or autosomal inheritance. And I call it all the ones that are Y-linked in this region blue. And so again, we see a lot of support based on pedigree data. This is indeed Y-linked. All right, so to wrap up then, color patterns have many heritable features, and we see that they have partially overlapping architectures, so they're partially separate, but we also have these genetic correlations between certain color traits. We find that these color patterns are highly polygenic, and we see that at least some of these traits are associated with copper number variation. And so what I'm currently looking into doing next is, do these C and V somehow actually make it easier to maintain variation? I think that could be really exciting. And so with that, I would like to thank my co-authors, especially Jocelyn, who did all the beautiful art and also took about 8,000 guppy pictures for me. And with that, I would love to take questions.
Well, thank you so much to the organizers for this opportunity. My research is grounded my research is grounded in the organism's natural history and motivated by the vast diversity of behavior that has evolved, specifically courtship behavior. As we see here, this male is desperately trying to find a mate. My research program addresses these two interlocking questions of how behavior evolves and how behavior affects evolution. And to address these questions, we use Drosophila, which sit at this unique intersection of a rich ecology paired with all the genetic and neural tools to actually dissect out the mechanisms underlying behavior evolution. And this is important because at the proximate level, behavior is controlled by neural circuits, and at the ultimate level, these circuits are patterned by genes. And what's really missing is understanding how evolution translates across these different levels of biological organization. And Drosophila provide an opportunity to fill in these missing gaps. The research I'm presenting today was my postdoc work, which was done in collaboration with a team of amazing scientists, and also wouldn't have been possible without these exceptional engineers who designed the high throughput equipment and analysis pipelines that I used. So what does mate choice look like in Drosophila? You may have noticed in my introduction that the male is reaching out and tapping all the individuals he encountered.
Every time we touch, I get this feeling.
And so in this moment where he reaches out and samples the pheromones of the individuals, he's assessing if it's the right species and if it's a female. And if he likes what he tastes, he goes on to vibrate his wings and sing a song, not quite as catchy as the one we just heard. But if he doesn't, if it's not the right individual, then he just walks away. And so my research focuses on this initial reproductive barrier about whether the male even decides to initiate courtship. I'm studying melanogaster and their closest relative, Drosophila simulans. They're about 2.5 million years diverged. And for the most part, the pheromones that the females produce are the same. But one pheromone that is different is called 711HD. Melanogaster females produce a very large amount of this pheromone, and simulant females produce a nominal amount. So when a melanogaster female is paired with a melanogaster male, he will court her. So you get a little taste of his song. But when a melanogaster female is paired with a simulans male, he has no interest in her. It's like, get me out of here. So I'm interested in, so this can be summarized in this data here, where melanogaster females are attractive to melanogaster males, but not to simulans males. Female simulants are attractive to males of both species. We'll see that males will court her eagerly. But what's very exciting is that we can take a simulans female and artificially perfume her with this melanogaster pheromone, and that is sufficient to almost entirely remove a male's interest in a female of his own species. And so I'm interested in understanding what is the genetics of this mate choice decision. How does this single cue result in such divergent behaviors? So going forward, I want to isolate the effect of just this pheromone. And so I will only be showing you data with simulans females that have been artificially perfumed with an increasing amount of this melanogaster pheromone. So over on the right is an unperfumed simulans female, and over on the left is still a simulans female, but perfumed with so much of the pheromone that it's equivalent to a melanogaster female. The y-axis is showing a courtship index, where 1 is no preference, anything above 1 is attraction, and anything below 1 is aversion. So melanogaster males, shown in blue, are essentially neutral to the addition of this pheromone, but simulans males, which is shown in yellow, are averse to the pheromone even at much lower concentrations than they experience in the wild. And so what is the genetic basis of this change? I took a candidate gene approach and decided to test the 2 transcription factors that are responsible for many of the sexual dimorphisms between species, which are known as fruitless and double sex. To test the function of these genes, I utilized the reciprocal hemizygosity test. This can be used for any trait of interest that is different between the species, as long as you can generate a hybrid and you can generate a null. So the idea is that if you generate a hybrid that is a null for one species copy of a gene, so in this case it only has a functional copy of blue C, if the trait changes, that shows that that gene is involved in that trait. And if you generate the reciprocal hybrid, in this case it only has a functional copy of yellow C. If the trait changes in the opposite direction, then that is very strong evidence that that gene has evolved for that trait. And this is very powerful because you can identify genes that have evolved because the genetic background is identical. So even if the gene is reliant on epistasis with another loci, you can still identify their effect. So in order to test these, I needed to generate nulls of these genes in the wild-type strains. And these have been traditionally difficult to target using CRISPR, and many of my first attempts were fruitless, pardon the pun. So during the process, we developed this new plasmid that is very easy for cloning and has high efficiency with CRISPR. It works not just in melanogaster, but in non-model systems as well, so it might be of interest to you. And in fact, it has such high efficiency that it's really changed the way I view the opportunities going forward. I'm working on ways to scale this test up from testing individual genes to a way to screen the entire genome. However, once I had these nulls, we could then look at the effect of these specific genes. So I'm starting by showing you fruitless, and what you can see here is that there's not any effect between the genotypes. So aversion to the pheromone is dominant, but all of the hybrids there's no difference in their preference. And we see a wildly different effect when we look at double sex. So here, the flies that contain melanogaster double sex are singing much more and at higher pheromone concentrations than the flies that contain simulans double sex. So this shows that double sex has evolved to affect pheromone preference. And when we compare these reciprocal hemizygotes, the dashed lines that each have one functional copy of double sex, to the hybrid, which has two functional copies, we see that there's a dosage effect as well. So that's indicating that this change may be due to a regulatory difference. One thing to note is these genetic backgrounds are identical except for which species copy of double sex they contain. So there's a very strong effect of a single gene, but it does not completely explain the difference because even at higher concentrations, the flies with melanogaster double sex do become averse to the pheromone. So there is at least one other gene involved, but there's a strong effect of this one gene. And by identifying the gene, we can then begin to better understand how this gene is functioning to change the neural circuit. So we do know a bit about the circuit underlying this pheromone preference. So when the flies are reaching out and tapping the female with their front leg, there are certain receptors that perceive the pheromone.
Every time we touch,
They then send the signal up to the brain with a set of ascending neurons that activate some general courtship neurons. And those same neurons indirectly inhibit the courtship neurons. And it's thought that the evolutionary change between melanogaster and simulants is occurring with a change in the net activation versus inhibition balance. And while fruitless patterns all of these neurons in the circuit, double sex only is expressed in these courtship neurons. So by identifying the gene, it leads us to a more specific subset of cells that we can study in order to understand its function. So you can imagine there are many different ways in which a gene could change a circuit. And the first thing I did was just look at the number of cells. So have there been a loss or gain of these specific double sex expression cells that's leading to the behavioral change? This is the fly brain up close. All of the green double sex positive cells are labeled here in green. They're a heterogeneous group of cells, and I looked at all the different subtypes and found that there's no difference in the reciprocal hemizygotes. So the number of cells has not evolved. And next, I want to see how the physiology or firing principles of these courtship neurons have potentially changed due to double sex. So to do this, we cut a little window into the head of a live male and put him on a microscope, and then measured the activity of the courtship neurons when presented with either a perfumed or unperfumed female. This video shows he reaches out and touches her, and then these are the set of neurons, and you can see that they light up and fire when he encounters this stimulus. And so we quantified this in the reciprocal hemizygote hybrids. And what is shown here is the flies with melanogaster double sex have a higher activity peak when they're exposed to the perfume. And the flies with the simulants double sex, the activity peak is the same for perfumed and unperfumed, but there's a faster rate of decay, so they're being inhibited and stopping the firing faster. So we looked at the same individual over multiple trials in a row, and then found that that pattern perpetuated and propagated, and when summarized over multiple individuals, this leads to this general trend of this higher activation of the courtship neurons in the flies with melanogaster double sex, and this faster decay indicating stronger inhibition, which is indicating that double sex is acting at that crux with these inhibitory neurons. So to investigate how double sex is leading to this change in activity, the final thing I'm looking at is how it is changing the wiring with these inhibitory neurons. So this again is the fly brain up here, and I've labeled the courtship neurons in green and the inhibitory neurons in magenta. And I'm hypothesizing that there's different number of synapses between them leading to this net inhibition. But it's very difficult to quantify that because the fly brain is very small. So I'm using this method called expansion microscopy, where you take a fly brain, which is very small, and embed it in a mesh, and then expand that so we can actually count the number of synapses, which are labeled here in yellow and blue, and quantify how double sex is changing the connections between these neurons that lead to the different activity and therefore the different behavioral output. So going forward, my lab is going to address more about when and where double sex is expressed to lead to these changes, what transcriptional differences double sex is causing, and how that changes the anatomy of these neurons to then lead to the behavioral change that we see. But on the other side of the coin, we're also going to be studying not just the variation between these species, but each of these species also has variation within them in terms of how much of the pheromone they produce and their level of attraction to it. So we'll be looking at using what we learn from understanding the mechanism in the lab and using a set of field experiments and field collections in natural populations and experimental evolution in outdoor mesocosms to test the relative role of natural selection and sexual selection of this complex behavior system. And so I think together these two complementary methods really provide this comprehensive picture of understanding behavior evolution from a holistic perspective. And just very quickly, I just started my own lab a few weeks ago. So right now there's me, a bunch of flies, some plasmids, and a very floofy mascot, but we're recruiting across all levels. So if you're interested, I think there's a little bit of something for everyone. And please contact me either at the conference or reach out by various media and questions afterwards. So thank you.
Thank you. And with that, I'd like to introduce Kathy Dara.
Hi, thank you so much to the organizers, Bernadette and Adam, for inviting me to speak today. I'm going to continue on the pheromone theme that we just heard about from Emily. So I think that studying chemical compounds that are important for communication is a really powerful system to look at evolution. And that's because organisms, we might be used to thinking about biodiversity, but there's a lot of chemodiversity or chemical diversity. Organisms produce all these different compounds. These compounds are important for fitness. This could be attracting a mate if you're a butterfly, or it could be pollinator attraction from flowers. And so I think this is a really great system to study evolution because we can measure these compounds quantitatively and identify the traits and test how they're important. So this is a mate choice session, so we're going to talk about sex pheromones. So sex pheromones are chemical signals that are important for mating. They could be involved in many different aspects of this, so it could be finding a mate or signaling mate quality, species identity. But today I'm going to talk about kind of a different type of pheromone, which is an anti-aphrodisiac pheromone. So these repel potential mates. They are transferred from males to females as part of a nuptial gift. And they, at least in the short term, could be beneficial for females by reducing harassment by other males when she's just mated to allow her to feed and lay eggs. And for the males, it ensures paternity. It ensures that the female won't remate, at least in the short term. And there could be some conflict about when this sort of bad smell should wear off, but I'm not going to get into that today. So the system that I was looking at this in is Heliconia's butterflies. I think that Rich Merrill in 15 minutes is going to try and convince you that these wing color patterns are very important, but I'm just going to ignore them and talk about the smell. So there's really good evidence that there could be these kind of anti-aphrodisiac compounds in Heliconias. In the 1970s, Mueller said that Heliconia's butterflies have a strong and nauseous smell. Kind of, I guess, pre-identifying chemical compounds, people tried to say, well, they smell like X plant or Y plant. A broken twig of an elder was someone's description. And these compounds that are really strongly smelling are found in the genitals, kind of glands of Heliconias. And really nice behavioral data has shown that these compounds indeed act as anti-aphrodisiacs. So males transfer the compounds to females when they mate. And if you take unmated females and paint her with these compounds, you can see that the courtship duration is reduced. And so males really are not interested in these females that have these nasty smelling compounds. And as I said, I think this is a really nice system because we can identify the compounds, quantify them, and in this case, in Heliconias melpomene, So what we're looking at here is time, so more volatile compounds elute earlier. And so you can see that there's this essentially one really volatile compound that's found in these mated females that's produced by males. And as I mentioned at the start, I think this is great that organisms produce all these compounds, but what I'm actually interested in is when different organisms produce the same compound, so convergent evolution. And in this case, this compound ocimene, which is the anti-aphrodisiac in these heliconius butterflies, is produced by both flowers and butterflies. So just as an aside, I think this is interesting because as part of the floral scent, this compound is attracting the butterflies, but I've also told you that it's acting as a repellent, an anti-aphrodisiac, when it's transferred from males to females. And so I think this gives us a hint at how complex these traits can be, because the chemicals themselves are not processed independently. There's other chemicals present, and there's also, of course, visual cues and behaviors that are involved. But today I'm going to talk more about the mechanism of how these different organisms are producing the same compound. So ocimene is a terpene. This is a class of chemical compound. And here are just a few examples of what is a terpene. So limonene is what makes lemon smell. Pinene is what makes pinecone smell. And linalool is what makes lavender smell. So we are familiar with these smells as sort of planty compounds. You might also have heard about terpenes because the cannabis industry is very interested in terpenes, as sort of different terpenes are thought to have different effects in these different strains. So when I found that I wanted to study the genetic basis of this, I was really excited because in plants, the genetic basis of terpene production is pretty well understood. So plants have these really large gene families of terpene synthases, or enzymes that produce terpenes. And so I thought, well, this is brilliant, right? We find this trait, we know how it's produced in plants, so we're just going to blast the genes and find it in the butterfly, and that's the end of the story. So what do we know about insects? Well, unfortunately, there are none of these terpene synthases in insect genomes when you search for them. And so this kind of, led to me questioning, well, are they even synthesizing the compounds themselves? If I've just said that the flowers are also producing the compound, so is it possible that they're just sequestering the compound from plants? But some previous work from another lab had found by labeling sugar with heavy carbon and feeding it to the butterflies, they found that this heavy carbon is incorporated into the ocimene. And so that means that the butterflies are metabolizing the sugar and making the ocimene themselves, not from a plant compound. So we do know that the butterflies somehow are able to synthesize this compound. But if they don't share these same enzymes, then how are they doing this? And so I then looked to the literature kind of from the past 10, 15 years, where in a few other insect species, the basis of terpene synthesis had been described. So how are they doing this? I know the biosynthetic pathways are, not the nicest thing to show, but I promise it's very simple. So there are these two very small molecules, and they're essentially sequentially added to form bigger and bigger molecules by these enzymes. And we know that this part of the pathway is present in insects because this is how insects produce juvenile hormone. And in plants, these compounds that are produced through this pathway are then converted to terpenes by these terpene synthase enzymes that I mentioned earlier. And in insects, they've actually co-opted this gene family that is usually involved in the elongation process and have turned it into a terpene synthase. So they've co-opted another part of the pathway to do this kind of second step. So what about the heliconius butterflies? Are they doing the same thing? So what we found was that there was duplications in a different gene family, but related, that's doing this third step instead of the second step, in the genome of Heliconius melpomene, which is this species that produces ocimene. And the reason why we thought that the duplications might be interesting is based on this theory that gene duplication allows for divergence in function. And so what we see is that in these Heliconius butterflies and also in Bicyclus, which also produce these terpene compounds, that there have been these lineage-specific expansions in these gene families that usually do the elongation process. And we can take advantage of the fact that these species differ in the types of anti-aphrodisiacs that they're producing to try and map these compounds. So what we did was we crossed these two different species that differ in their anti-aphrodisiac production. Heliconia cydno on the left doesn't produce ocimene, Melpomene on the right does. We can then use these to carry out quantitative trait loci studies. So we analyze the phenotype to quantify the amount of ocimene production. We then do low coverage whole genome sequencing to get the genotypes. And we combine this information to try and identify regions of the genome that are associated with ocimene production. And what we find is that there was a significant association between ocimene production and the genotype on chromosome 6 of these butterflies. And thankfully, the gene family with the duplications that we were interested in is also found on chromosome 6 within this significantly associated region. But there's still 6 genes in this gene family, and this is a lot to test functionally. And so we turned to RNA-seq data to see if we could find a way to narrow down our list of candidates. And so what we were looking for is genes that are most highly expressed in the male abdomens, because that's where we know that these compounds are being produced. And we found that one of these genes followed this pattern. HMLOS was most highly expressed in the abdomens of males. And so then we decided that to really show this, we had to get the function of the protein. And so this gene encodes an enzyme. We expressed it in E. coli. And then we assayed the enzyme activity. And we were able to do this because, as I said, we know the biosynthetic pathway. And so we know what the substrates could be for the enzyme. And then we identify what the compounds are that are produced by the enzyme. And we find that when provided with the correct precursor from this pathway, that this enzyme is able to convert the precursor to the anti-aphrodisiac ocimene. And so it's acting as a terpene synthase in these butterflies. There's also been slight follow-up to this by a master's student in the lab who worked on this divergence between closely related species. So, as I said, Heliconius melpomene produces this ocimene, Heliconius cydno doesn't. But you might have noticed that it's not that Heliconius cydno doesn't express any of the protein, so the protein is being expressed here. But when we express it in E. coli, this orthologous gene in the other species is not able to produce ocimene. So we think that it's due to these coding differences in amino acid changes in the protein itself, rather than that it's just been downregulated in this other species. And so insects have actually co-opted different gene families for this new function of terpene synthesis. And so this is a really nice example of an independent and repeated evolutionary origin of a novel enzyme function in different insect families. So again, we have these plant terpene synthases, which aren't related to the insect ones. And in these beetles and other insects, they've co-opted one of the, we'll call it number 2 of the elongation gene families. And then in Heliconius, they've co-opted this other gene family to carry out this same function. But what I'm interested in working on now is the fact that butterflies aren't the only insects that produce ocimene. And so bees, both bumblebees and honeybees for different purposes, produce ocimene, as do a bunch of other insects. And so I think that this is a really great system to look at convergent evolution. I think often when we talk about repeated evolution of traits, we're talking about similar but not identical traits. Whereas when we're talking about compound production, the trait is identical. It's the same isomer, it's the same compound. We can quantify it, we can quantify the efficiency of the enzymes at producing these compounds in different species. And so I hope that this will allow me to carry out experiments to work out exactly what amino acid changes are involved in the evolution of this enzyme function across the different insect families. And so with that, I just want to thank everyone that helped with this work, funding, and also that I've just started at Indiana University in January. And so if anyone is interested in insect pheromone biosynthesis and its evolution, please come chat to me. Yeah, yes, there is. So within Melpomene, there's, I would say, quite a big amount of variation. So they all individuals have ocimene, but some have lots and some have much less. And so that's also something that's really interesting to think about. Kind of, are females choosing males that have more of this compound because it gives them better protection against harassment, for example? Like, that's really hand-wavy, but you could imagine that it plays a role in female choice. But we don't know.
Thank you, Kathy. Okay, now I'd like to introduce Richard Merrill.
Hi, great. First of all, thank you for the organising the symposium. I'm really excited to be here. Got big acts to follow. I hope I can do my best. So I'm going to talk about some work that we've been working on in my lab for the last 7 years or so, and something I've been working on for approximately about 15 years ago. 15 years or so. But really, this work has been done by two of my PhD students, Matteo Rossi and Alexander Hausmann. And they should get the credit. But of course, we've done this in collaboration with a lot of people, particularly at the University of Rosario. So, like many people in my lab, we're interested in diversity, we're interested in speciation, which relies on the accumulation of genetic differences that cause or result in barriers to gene flow between closely related organisms. But what's often not realized is that many closely related animals remain separate not because they can't interbreed, because they have sterility and viability, but because they effectively choose not to. And this often depends on visual cues and visual preferences. If you don't believe me, just consider our own species and wonder why we don't have more chimpanzee-human hybrids. It's not because we have different numbers of chromosomes, or not primarily. So visual preferences are probably important. Visual signals are important for speciation, but we really know very little about the genetic basis of these situations, especially the preference. So this is what I've been studying for a number of years in two species, Heliconius sydno, which I shall try to refer to as the white butterfly, and Heliconius melpomene, which is a red butterfly. And these are very closely related and sympatric in Central America, where I spend a lot of time doing this. They clearly have very bright patterns. One is white, one is red, and these are under divergent selection for mimicry because they're both distasteful, they're warm predators. But these patterns are also involved in mate recognition. So males of the red species prefer to court and try and mate with females of the red species, and males of the white species prefer to court and try and mate with females of the white species. And this leads to quite strong assortative mating between these two species. So what about the genetics of this system? So the genetics of the color pattern switch is actually relatively simple. So we go from the color pattern of the white butterfly, Sydno, to Melpomene. There's really just three major loci that underlie this color pattern switch. And we know what these genes are, and to some extent, we're beginning to understand how they act. I'm just going to point out this one gene here on chromosome 18 called optics, because it's important later. And this is what causes this switch from red to white on the forewing, which is probably the major visual cue in the mate choice. What about the mating preference? So back in my PhD, I spent a lot of time doing QTL mapping. We cross these two species, you can. So effectively, you're shuffling the genes from each of the two species, and then we tested hybrids in fairly simple mate choice assays. So we put male hybrids into a cage with a virgin female of each species, and we count the number of courtships directed towards each. And just reducing about 5 years of my life to 3 arrows, we found three major effects, loci on chromosomes 1, 17, and 18. And the one on 18 is really nice for two reasons. First of all, it's a very large effect. It seems to explain about 30% of the difference that we've measured between the two parental species. And it also happens to be in almost exactly the same location as one of our major color pattern genes. To the extent that if you have an allele from the red species here, you're more likely to court the red females than your brothers that have inherited two alleles from the white species. But you don't actually need to do any genetics, because we can determine the genetic loci simply based on color pattern, because the genes are so tightly linked. So this suggests that there's an allele underlying the color pattern switch, or a gene underlying the color pattern switch, and a gene underlying this preference change, which are at the very least very tightly linked in the genome. And this has nice consequences for speciation. And that was great. But QTL are regions, and really the key question, or one of the questions we wanted to ask, is what are the causal genes underlying this? And this is an easy question to ask, but a rather difficult question to answer. And that's because, as I said, QTL are regions. So we take our QTL confidence region at the end of chromosome 18. So this is the region where we kind of can realistically expect to have a causative mutation. And this includes our QTL peak, which is just the region where we have the strongest statistical support for blockers. There's about 200 genes. So how can we go from a QTL to a gene or genes affecting this preference? So what we did is we started to take a population genomics approach. So previously I suggested that Cydno, the white butterfly, and Melpomene, the red butterfly, were sister species. This was a lie, because there's another species called Heliconius timeretta, which is more closely related to the white species, and in fact it's its geographical replacement to the east of the Andes, but it looks more like, or it has the same color patterns as the red species Melpomene. And we know the mechanism underlying this is due to adaptive integration of optics alleles from Melpomene into timeretta. So there's been some past hybridization event which has allowed these alleles to kind of jump into the other lineage. And our thinking was, well, if adaptive integration is responsible for this new color pattern, perhaps adaptive integration has also been responsible for differences in preference, because we know the white species prefer the white, and this new timeretta also prefers the red species. And if this is the case, there's three quite nice predictions we can make. The first is we might expect to see increased allele sharing at the QTL. I'm just going to have some water. Okay, so what do I mean by that? Well, we should just expect to see evidence of gene flow at this particular region of the genome. So what we did was we used a statistic which is based on the Abba-Baba test. If you understand the Abba-Baba test, well done. But it's relatively simple, and it's really just looking at differences in patterns of shared and derived alleles to look for differences between, for situations where we have different genealogies due to incomplete lineage sorting, as opposed to those which might be due to incomplete lineage sorting or gene flow. And the basic point is that we can use this statistic thought up by Simon Martin, FD, and use sliding windows across our region of interest, like so forth. Low values of FD suggest that this region of the genome is following the species tree, so Cydno and timeretta are clustering together with Melpomene as an outgroup, but high values of FD suggest that there's been gene flow within this region. So when we look at this region, hopefully you can see that there's two fairly nice peaks. One is at the regulatory region of optics, our color pattern gene, and we know from past evidence that this has been acquired through adaptive integration. But we also see a nice peak right within our QTL peak. So right within this region of the genome where we know is strongly associated with our visual preference change. And actually, when we look at patterns of, or kind of signatures of integration using other methods, we find a peak in exactly the same region, and then we look at divergence between our white and our red preferring species, we also find a peak of divergence. So this just does suggest that there's probably been some integration at this point of the genome. But that doesn't necessarily mean it relates to our behavior. So the second prediction is that we should expect to see coincident preference QTL. So what do I mean by that? So previously, we've been able to show that this region of the genome affects differences in visual preferences between Cydno and Melpomene through my QTL studies. If there has been adaptive integration, we should expect the same thing, or the same region of the genome, to affect differences in preference between Cydno and timeretta. So what did we do? Well, we basically just recreated what I did during my PhD. We generated crosses, but this time between timeretta and Cydno. We did a lot, or not really me, Lena, Danny, and Geraldine did a very large number of mate choice trials. And we just looked to see whether the preference was segregating with this red color pattern, was segregating with this region of the genome. So the prediction is that if this region of the genome also affects mating preference, individuals which have the red pattern, so inherited an allele from the red butterfly and the white butterfly in these back crosses, should have a stronger preference for red females than their brothers that have inherited both alleles from the white. And of course, I wouldn't show you this unless this was exactly what we found. There's a lot of noise because this is behavior, and these are heliconias. But we do see a very clear difference in the behaviors of these two butterflies segregating this gene. So two kind of nice predictions fulfilled. The third prediction is that we might see shared patterns of gene expression. Of course, phenotypic differences can be due to other reasons, like protein coding differences, but they may be due to differences in gene expression. So we looked at neural gene expression in the brains of these butterflies, and we had previously looked at our original pair, Sydno and Melpomene, and within our candidate region, we'd found 27 differentially expressed genes. This is quite nice, but 27 is still quite a large number. So we decided to look more broadly, this time including our third species, Timoretta. And actually, we looked at multiple populations of Sydno, Timoretta, and Melpomene to allow us to have multiple comparisons. And what was rather nice is that when we did this, we found just one differentially expressed gene within our candidate region. It's a gene called regucalcin. Don't worry too much about that. What I think is more important is that it's upregulated in Heliconia sydno compared to the red-preferring butterflies. And even more excitingly is where this gene was, because it happened to be right within our QTL peak, right within kind of this region where we have the strongest statistical support for a preference gene, and also right under this peak of integration. So we were pretty, well, I was pretty pleased. So what can we do with that? So one of the things I often got thought, what have you thought about CRISPR? Because CRISPR solves everything. Except it doesn't really, because CRISPR is a wonderful technology, but in butterflies we can only do certain things at the moment. And we can really just knock out genes. And this is a relatively crude tool. It works very well for color patterns because you can knock out a gene and see if the color pattern is disruptive. But if you think about it, what we're really doing is we're taking an organism and we're trying to break it, and we're trying to break it in a sensible way to suggest that this gene affects our phenotype. If you do this for a behavior, if you break a butterfly, it's unlikely to behave normally. Nevertheless, Matteo, my PhD student, really, really wanted to do this, and I thought, well, go ahead, Matteo. Done quite well so far. Try it. And he did. He knocked out regucalcin, so we basically deleted this region, this part of the regucalcin between exon 1 and exon 2, and we generated a number of knockout individuals. I'm not going to go into this experiment in too much detail because I don't have time, but basically what we have two treatments. We have our mosaic knockouts, because these are mosaics, where we knocked out regucalcin, and then we had non-deletions, which are basically individuals, where we did the CRISPR experiment, but it didn't work. And so the prediction is that in our knockouts, they should court less. And again, this is what we found. We found that when we knocked this gene out, they courted females less. As I said, we're just really breaking the butterfly. So what we weren't sure about, whether this was just that it was actually affecting courtship, or whether it was just affecting the butterfly in general. So in these same individuals, we recorded other behaviors that presumably rely on vision, particularly feeding and flying, and we found no difference between our two treatments. So this really links our gene of interest to our phenotype that we're interested in. So just a kind of quick summary. Q and preference in these two butterflies seem to have a relatively simple genetic basis. There are obviously other genes involved, but there are major effect genes underlying both components of sexual isolation. Regucalcin seems to be a visual preference gene, and we think it's probably one of the first visual preference genes that's been identified. But more importantly, it's different to the optics gene, which causes the red-white switch itself, so they can evolve independently. And it can play a different role. So when we have our two red butterflies, it probably is involved in convergence in some way. but when you have a white and a red pair, say in Panama, it seems to be a barrier to gene flow. And colleagues in Konrad Lose's lab have shown this. And then also we think that this quite nicely demonstrates that adaptive integration or hybridization can be important during the evolution of behavior. I just finished by thanking Matteo and Alex and all the people in my lab and many of my collaborators, and just say there's a lot of work to be done. I'd like to work out what this gene does. We have some information, but not too much, and we're always looking for people to join the lab. And we're in Germany. We make BMWs. We have money to At the back, hi.
Do you know the directionality of the adaptive integration? Was it from
We don't know. What makes most sense is it from Melpomene into Timaretta, so from our original red butterfly into the white-red lineage. There's a selective sweep at the same point in Timaretta, but not in any of the other butterflies, which really kind of suggests that's the receiver. But I think we're not 100% sure. It kind of makes more sense in my mind, but it's kind of hand-wavy answer from it. Hi, Amanda.
What kind of gene is it? Is it transcription factor? Is it?
We don't know too much, so they're pretty common. It's meant to regulate calcium, but we really don't know. We've done antibody staining in the brain, and it's expressed in the kind of the optic lobe and in the brain, but it's pretty broadly expressed, and we really don't know. It's a kind of unsatisfactory answer, and it's kind of what I want to find out, but I suspect it may take the rest of my life. Okay. Thanks very much.